All right, today we're going to be detectives. We're going microscopic, peeling back the layers on a single, hidden culprit that's behind some of the toughest diseases out there.
So think about this for a second. What could possibly link something like Alzheimer's with heart failure or kidney disease? I mean, they seem totally different, right? One attacks the mind, the others attack our organs. But what if they all share the same destructive secret, way down at the molecular level?
Well, that's exactly what's going on. The connection is this one single microscopic problem. Just a tiny, tiny malfunction that has absolutely devastating consequences.
And here it is. The culprit's name is amyloidosis. Now, what's wild about this is it's not some invader like a virus or bacteria. Nope, this is more like a civil war inside our own bodies, where our own proteins, the very things that are supposed to build us up, go completely rogue and turn against us.
So how in the world does that even happen? Well, to get a handle on amyloidosis, you first have to understand the core issue. It's all about our proteins behaving very, very badly. Okay, check this out. Think of a normal, healthy protein like a perfect little piece of origami. It's got this super specific, complex shape, and that shape is everything. It lets the protein do its job. But with amyloidosis, that protein just misfolds. It's like instead of that perfect origami crane, you get a crumpled, sticky wad of paper. And this thing isn't just useless, it's actually toxic, sticking to all the other crumpled wads around it. And that clumped up, sticky gunk, that's what we call amyloid. Now, the key word here is insoluble. That's what makes this so dangerous. We're not just talking about messy clumps. These are incredibly tough, little thread-like things called fibrils, and our body has no idea how to clean them up. It just can't dissolve them. And these fibrils have some really unique and really dangerous properties. They've got this special structure called a beta-pleated sheet, which basically makes them super stable and rigid, kind of like microscopic rebar. And that's why the body can't get rid of them. They're just too tough for our natural housekeeping systems to break down. So what happens? They just build up, and they get in the way and start messing up how our tissues are supposed to work. Okay, so we've got these tiny, nearly indestructible fibrils building up. But how do they go from being a microscopic nuisance to causing massive damage? I'm talking full-on organ failure.
It's basically a catastrophic chain reaction. It starts small, with just one protein folding the wrong way. Then, more of them clump together, forming those fibrils we talked about. And then, these fibrils start getting deposited in the spaces between our cells, inside our organs. As they build up over time, they literally start crowding out the healthy cells. They choke off the supply lines for nutrients and oxygen. The organ gets stiff, it can't function properly, and eventually, it just fails.
And to really wrap your head around this, you have to appreciate just how small we're talking. I mean, look at this. A single one of these amyloid fibrils is only about 7 to 13 nanometers wide. It's almost impossibly small. To put that into perspective for you, that is thousands and thousands of times smaller than a human hair. It's a genuine microscopic villain. But here's the kicker. When these tiny little things pile up by the billions, they have enough power to bring an entire organ, like your heart or your kidneys, to a complete grinding halt. All right, let's zoom out a bit. Because this one core problem, this protein misfolding issue, it doesn't just cause one single disease. Oh no, it's the engine behind a whole gallery of different conditions. See, the key thing to understand is that the type of disease you end up with all depends on two things. First, which specific protein decides to go rogue and misfold, and second, which organ it chooses to build up in. Just take a look at this. For Alzheimer's, you've got a protein called amyloid beta, or A-beta, that builds up in the brain. But then you have a type of amyloidosis that's linked to multiple myeloma, and that's a completely different protein, immunoglobulin light chains, or AL, and it targets the heart and kidneys. Then there's ATTR amyloidosis, where a protein called transthyretin gums up the works in the heart and nerves. See the pattern? It's the exact same mechanism, just a different protein and a different target, and a different tragic result. So let's pull all this together. What is the single most important thing to take away from all of this? The core idea. It's this, right here. The issue isn't about having too much protein, it's about having the wrong shape of protein. This isn't a disease of quantity, it's a disease of quality, or maybe, more accurately, a disease of structure. The body isn't necessarily overproducing anything, it's just that the proteins it is making are folding into these toxic, dangerous shapes. This quote really just nails it. The whole underlying issue is the misfolding and aggregation of structurally unstable proteins. It's a breakdown of structural integrity at the most fundamental level of our biology. You have these unstable proteins, and they just collapse into a dangerous new form.
And that leaves us with a pretty fascinating and maybe even hopeful final question to think about. We know the problem is protein misfolding. So what happens if we can finally crack that code? What if we could figure out how to stop proteins from misfolding in the first place? Or maybe clear out these clumps before they do any real damage? Just think about what we could achieve. It's a question that could literally change the future of medicine.